Hello. Welcome to World of Microbes. In today's video we are gonna know about zeihl-neelsen stain, its objective, principles, reagents, preparation of reagents, procedure, results and interpretations, applications and limitations. Do like. The zeihl-neelsen staining technique is a differential staining technique that was initially developed by Zeihl and modified later by Neelsen, hence the name zeihl-neelsen stain. Zeihl-neelsen, ZN, staining is a bacteriological technique used to identify acid-fast organisms, particularly mycobacteria. It's used to diagnose tuberculosis, TB, and other infections. Neelsen used carbon flushing from Zeihl's experimenter, which is a basic dye and chemical compound that is used for staining bacteria, with heat, and added a decolorizing agent using acid alcohol, a differentiation reagent and a counterstain using methylene blue dye, thus developing the zeihl-neelsen technique of staining. The use of acid alcohol in the technique earned it the stain, and the application of heat in the technique gives it the name the hot method of acid-fast staining, which is a synonymous name for the zeihl-neelsen staining technique. Do like and subscribe. Wondering why use this technique instead of gram staining or negative staining? Well, because this technique is used on microorganisms that are not easily stained by basic stains such as negative staining or gram staining. Do like and One of the most complex microorganisms that require harsh treatment of the zeihl-neelsen compounds is the Mycobacterium spp. Mycobacterium, actinomycetes, Norcadia, Isospora, Cryptosporidium, and some fungi contain a thick cell wall made up of lipoidal complexes known as mycolic acid, which make the bacteria less susceptible to. These molecules also help the bacteria to subvert and then hide from the immune system. Mycolic acid is difficult to stain, and therefore simple stains like gram staining cannot penetrate the thick cell wall of these organisms. They require harsher treatments to allow stain penetration for identification and examination, and hence the use of the zeihl-neelsen or the hot method of acid-fast stain. So, the objectives of the zeihl-neelsen staining is to differentiate between acid-fast bacilli and non-acid-fast bacilli, and to stain Mycobacterium species. Do like and subscribe. Acid-fast bacilli refers to bacteria that retain a red stain after being treated with acid alcohol during a staining procedure, meaning they are resistant to decolorization by acid. Acid-fast bacteria have a thick, waxy cell wall containing high amounts of mycolic acid, which allows them to resist decolorization by acid alcohol during staining, while non-acid are bacteria that readily lose the stain when exposed to acid alcohol, appearing decolorized. The most common example of acid-fast bacilli is Mycobacterium tuberculosis, the bacteria responsible for tuberculosis, TB. Do like. Principle of the zeihl-neelsen staining is, the zeihl-neelsen stain uses basic fuchsin and phenol compounds to stain the cell wall of Mycobacterium species. Mycobacterium does not bind readily to simple stains, and therefore the use of heat along with carbon flushing and phenol allows penetration through the bacterial cell wall for visualization. Mycobacterium cell wall lipid content made up of mycolic acid on its cell wall, making it waxy, hydrophobic, and impermeable. These are SS-hydroxycarboxylic acids made up of 90 carbon atoms that define the acid fastness of the bacteria. Use of carbon flushing, which is basic, strongly binds to the negative components of the bacteria, which include the mycolic acid and the lipid cell wall. Alcohol along with the application of heat forms a strong complex that cannot be easily washed off with solvents. The acid-fast bacilli take up the red color of the primary dye, carbon flushing, while non-acid-fast bacteria easily decolorize on the addition of the acid alcohol and take up the counterstain dye of methylene blue and appear blue. This technique has been used in the Bacterium tuberculosis and Mycobacterium leprae. Do like and subscribe. Now let's talk about the reagents used and their preparation in the zeihl-neelsen stain. Do like and subscribe. Reagents used in the zeihl-neelsen stain include carbon flushing, primary dye, 20% sulfuric acid or acid alcohol, decolorizer, methylene blue dye, counterstain, or malachite green. Now how these reagents are prepared. Carbol flushing, distilled water, 100 ml, basic flushing, 1 g, ethyl alcohol, 1 mol, 10 ml, phenol crystals, 5 ml, acid alcohol, 3% hydrochloric acid in 95% ethyl alcohol, 95 ml, distilled water, 2 ml, concentrated hydrochloric acid, 3 ml, 0, 25% methylene blue and 1% acetic acid, 0, 25 g, Distilled water, 99 milliliters. Acetic acid, 1 milliliter. Do like and subscribe. On a clean sterile microscopic slide, make the smear of the sample culture and heat fix the smear over blue heat. Over the smear, pour and flood the smear with carbol flushing and heat gently until it produces fumes. Allow it to stand for 5 minutes and wash it off with gently flowing tap water. Add 20% sulfuric acid and leave it for 1 to 2 minutes. Repeat this. Appears pink in color. Wash off the acid with water. Flood the smear with methylene blue dye and leave it for 2 to 3 minutes and wash with water. Air dry and examine the stain under the oil immersion lens. Do like and Now about results and interpretation. Acid-fast bacteria retain the primary dye, carbol flushing, and stain pink. Non-acid fat bacteria take up the methylene blue dye and appear blue. Do like and Applications of zeihl-neelsen staining. Used for examination and identification of Mycobacterium species. Used to differentiate between acid-fast and non-acid fast bacilli. Used for the identification of some fungal species such as Cryptosporidium. Do like Limitations of zeihl-neelsen staining are, it can only be used to identify acid-fast bacilli, and the physical morphology of the organism is distorted. Do like and subscribe.